All right, welcome everybody. It's my great pleasure to have Professor Michael Marty today give the lecture. He's an expert in native mass spectrometry and developer of a pretty awesome tool, UniDec, that some of you may have seen already as we use it in the lab quite frequently. And yeah, he's gonna give us an intro to native mass spec and in particular the use of UniDec. And before he gets started, maybe a little background about him. So he's an analytical chemist. He did his undergrad at Olaf College before he then moved to the University of Illinois at Urbana-Champaign. And then he did a postdoc in Oxford at the University of Oxford, and then launched his own lab at the University of Arizona. I think he's meanwhile an associate professor, so walked up the ranks quite fast. No wonder with like all the awesome stuff they do. And yeah, I'm very much looking forward to your lecture.
Awesome. Thanks, Daniel. It's a lot of fun to be here today. And just to give you kind of an overview, I have about 10 slides prepared. So we'll do just like a quick intro, focusing on mostly like how UniDec works and like a few of the high level overview features. And then the rest of the time, Daniel sent some example data. So we'll play with that. And I have like, we can demo anything you want to see. So like, we can just kind of go through it. And part of this will be a little bit of a product launch. So we have a new module that's coming out in version 6 that we're just about ready to release. So you get to see that, I think, for the first time of anyone in public, which would be cool. So anyway, let's get started. So, you know, I guess a lot of you come from the small molecule world. I definitely come from the opposite side in the large protein world. And you know, in the small molecule world and the protein world, the problem is the same with mass spectrometry. We have mass over charge, we need to figure out mass. And for small molecules, it's relatively easy because you can measure the charge state by the isotope distribution. And for large molecules, we measure the charge state typically by the charge state distribution, although sometimes by the isotopes as well. And so most of you probably learned this when you took analytical chemistry or mass spectrometry, that you can basically solve this equation where if you have two peaks and measure the mass over charge and you know they're one charge apart, you can solve for the charge state. And from here, then you can calculate the mass, right? And so this is how people, you know, have done this for a long time in the field, either with spreadsheets or with calculators. But I would say more recently, starting with Maxent and then a number of other different algorithms that have been developed, we've tried to figure out ways to solve this with computers and make basically direct deconvolution possible. Because, you know, like in a spectrum like this, it's relatively easy to pick out the charge states. In a spectrum like this, it's really hard to understand what's going on. And so, you know, we have lots of different peaks, a lot of them overlapping. We need ways that a computer can solve this for us. And so that's where we developed UniDec as a way to kind of solve this problem of having multiple charge and mass states all overlapping. And so the advantages of UniDec is that it's fast, it's free, and it's open source. It's now up on PyPy, so you can just pip install UniDec and get it on your computer. It's automated but also interactive. So we have a GUI so you can play with it and kind of interact with your data. It's, you know, like as much as we would love to have it fully automated, there's still a lot of room for users coming in and manually helping to interpret, especially as the data gets more complex. We can get a lot of quantitative information on the mass, on the intensities, on the charge. And the really nice thing about UniDec that I would say distinguishes it from other algorithms is that it's built to work equally well for simple and complex spectra. So it'll work just as well for that really simple distribution as it will for something super complex like this. And really the speed and the scaling just go by the size of the data and the number of charges that you allow. We also, because the algorithm underlying UniDec is very flexible, it can deconvolve in multiple dimensions. So we can bring in things like ion mobility information. We can bring in CDMS. We haven't really made use of this for chromatography data. We more just sort of gone scan by scan, but in theory, that could also be brought in as well. And as the tool has grown over the years, it's expanded to include a lot of different auxiliary tools for native MS analysis. It's gotten a little out of hand, if I'm honest, but there's a lot in there that you can use if you know what you're doing. So I want to briefly go over how the algorithm works. We have a spectrum that we get, which looks something like this. And of course, these are all different charge states. What we're going to initially assume is we're going to assume all possible charge states are equally probable. So what we're going to do is basically take this one-dimensional spectrum and convert it into this two-dimensional space of spectrum versus charge. So mass over charge versus charge. And the goal basically is then to assign for each of these data points what the charge distribution is going to look like. And in a simple thing like this, it really should just be one charge state per data point. But in more complex overlapping distributions, this of course can be multiple charge states. And it's trying to solve this matrix here. And the way that it does that is by looking at each data point. So say, let's take like this one here, and then it looks for neighboring charge states. So it'll say, okay, go up a charge, look at the intensity for that particular value, go down a charge, look at the intensity for that value, and try and have a smooth distribution. It actually uses like an exponential, sorry, it's a log mean filter. So it's going into log space, and then it's looking for like basically like a linear distribution in log space. So the first step is that it's going to try and smooth out the charge distributions. The second step is that it basically is going to go in and say, well, assume that if we have a spot here, a data point here, that we have some sort of peak shape distribution around it. And then it's going to create a template spectrum, basically by summing together all of these peak shapes for all of these different charge states. And then it's going to match that with the data and say, well, this one's not quite high enough. I need to boost that intensity. And then it's going to repeat that whole process. So basically, you know, we're smoothing the charge state distribution. We're creating a template spectrum, and then we're going to match it to the data and then repeat. And so by doing this for about 10 to 50 iterations, it'll eventually converge. And what we're left with then is an output that looks something like this. So we have mass over charge and charge, the same sort of grid that we have here. And each of these spots now represents sort of the charge distribution. And so this is like the raw output of UniDec is just this mass over charge versus charge. It's then going to transform that from mass over charge into mass so that all of those spots now line up and you get the mass distribution. And of course, we can sum it along the mass axis to get the charge distribution. We can sum it along the charge axis to get the mass distribution. And that's mostly what we're going to look for, is that summed mass distribution. Any questions on this? I realize I went through that pretty quickly. I realize that's a lot of math to handle. It's all published if you want to go back and look at the original papers. Actually, each of these steps is optional. So you can choose whether or not you want to do this charge smoothing. You can choose whether or not you want to have a peak width. There's a lot of like ways that we can play with this algorithm. Okay, no burning questions. I'm going to keep going then. Okay, one other feature, we'll now get it. That's just sort of like how the base works. And so I want to go into now some of the features. While we're talking about the algorithm, I want to highlight something that not many people know about. It's actually been in there for a few years now. It's a feature called double deck. And basically the problem was we were doing these experiments. And this is something I think would be potentially useful for what you all are working on. You know, like we have this experiment where we have a complex natural protein. So this is bovine rhodopsin. It has a series of glycoforms. It has different post-translational modifications in addition to the glycoforms. We wanted to look at zinc binding. And so like, this is the holopeak, this is the zinc bound, but then these are all like different glycoforms, but they also have different zinc bound states. And so what we wanted to do was basically say, could we deconvolve the deconvolved spectrum so that we put in like a template spectrum and have it basically collapse that into a single point. And so basically what we can do like this. So basically like take this spectrum and end up where instead of having this complex distribution, we just have a single peak for the holo and a single peak for the zinc bound. And so the way that we did this is you feed in here, we just took like the protein, we treated it with EDTA. So it stripped out all the zinc. And we basically said, this is the intact distribution over here. So you can see we've lost the zinc bound peak. We've lost a few of these minor peaks up here that are basically the PTMs plus zinc. And so when you feed in this template, you can basically double deconvolve it and get out this, which would be just like a totally clean spectrum. And when you add in zinc, this is what you get. And you can use this even on very complex distributions. So you can see like, here's the distribution of zinc bound to the holo, and this is with the first glycan and the second, and it just collapses it all into this. So it's a way to basically automate like analysis of very complex spectra when you have a template and you want to see what's binding to that. So all you need to do is like turn on double deconvolution, select the template, and it'll do the rest of it for you. And so it's useful for looking at like binding to complex samples. So yeah, that's all built into regular UniDec. So this is the core base package. This has been around since 2015. So it's I've been working on this for I guess 8 years now, which is kind of fun. The basic UniDec does 1D spectrum. So you put in like a single spectrum, it deconvolves it. Lots of tools are built on this. You can do batch processing where you select a series of files. If you open up a raw file, it's just going to collapse it and average it into a single scan. There are ways you can do ion mobility. So if you switch and use into ion mobility mode, you can do this now. You can see this is arrival time versus mass overcharge, and you end up with these crazy 4-dimensional data sets. And you can do that as well. It's not as well developed. It's not as smooth. So I'm sorry about that, but it does work and you can try it out. Another useful tool is MetaUniDec, which we developed in 2017. Basically, the idea with MetaUniDec is that you can layer a series of data in like a series of spectra. So you could say, if you're doing like a collision voltage ramp, you could layer each of the collision voltage steps as a set of data, and you could extract different sorts of information from there and fit the different curves in different ways. And then from MetaUniDec kind of was birthed Unichrome, which I was talking with Daniel about briefly earlier. This is basically our take on how you look at chromatography data. So you can see here we have a total ion chromatogram, and we can go through and either manually select regions of the tick, or we can automatically parse them in different ways by either like picking chromatogram peaks or by just like sliding window or a regular window. And basically it's built on the same MetaUniDec backbone. It just like slices the chromatogram in different ways, generates all the different spectra, and then you can deconvolve this all at once and either like export it to regular UniDec or do some manual things. I'm particularly excited about being able to do this. So this is doing like a extracted ion chromatogram for the intact masses that come out, because we're basically getting mass versus time. Anyway, and this will support Waters, Thermo, Agilent, MZML, and actually MZXML now, I think, although I'm not totally sure I've tested MZXML with this. So yeah.
This is super cool. And as we were very interested in that,
yeah.
Can you do feature finding with that and then export like a peak table out from this data?
So you can definitely do like peak detection. It's maybe not the most sophisticated. The advantage though is that all of the data is there. So you can export like a grid of mass versus time, right? And then, use whatever sorts of tools you want. And if you have ideas for ways to build on this, it's all open source and we love for people to play with it. Like all the data is there. So like you can do anything you want with it. And actually it's all stored in this HDF5 file, which is a nice open format that you can read in. So yeah.
That's cool.
I would say there are rudimentary tools for doing it, and we can play with that and take a look at it. But yeah, cool. Any other questions on this? We can definitely take a look during the demo. Cool. So the other exciting thing that we're working on, one more slide before that. Last year, I guess a year, maybe more than a year ago now, we introduced UniDec CD, which is UniDec for CDMS data. And so I'm not sure how many people are aware, but you can use Orbitraps for doing CDMS, charge detection mass spec, where you measure like single ions, and by looking at the intensity of the ion, you can say something about the charge. And so you get data that naturally looks like M over Z versus charge. So this was just perfect for UniDec because that's actually the format that it was built for. And so it'll now read data, and then you can actually like not just read data and deconvolve it, but you can then like apply the same UniDec algorithms to sharpen up those peaks. And so like naturally the data actually has a fairly large charge spread, and that's what leads to all these different satellite peaks. But when you apply UniDec, you can smooth it up and you get this one really nice peak. And I think we were pretty excited. To our knowledge, this was the first like real software developed for CDMS at all. Everybody else had been using sort of custom scripts and things. And so this gave a nice user interface that's open source and that other people can develop. And so we've tried to support, it definitely supports thermo data in a variety of formats. But even people using home build instruments, if you just put in like a CSV file, it'll open it up and you can do the same sort of things. So you could do this on your Q Exactive actually. It's not too hard to do. You just have to like tweak a few settings and you're down in CDMS mode, which is cool. So, and then the last thing that we've been working on is this is the new thing we're about to release in UniDec 6. We call it the UniDec processing pipeline, or UP. And the idea is that, in pharma companies or other settings, you end up with like a giant Excel list of like, here's all my runs, right? I ran a 96-well plate, and they want to go through and say, here's all the different masses that I expect. Did I get the correct masses? Or here's the masses that are incorrect that I'm worried about. Did I find those? And so basically this will take in an Excel file. I had to basically cobble together a version of Excel inside of WX Python, so that you can just sort of drop and copy paste things from Excel into here. But then you just feed it your sample name, and I'll show you some of the other things like the directory. And then you feed it the sequences that you want, and then you tell it which things you're looking for. So it can be like the correctly paired, maybe like 1, 2, 3, but then you might also be worried about homodimers or different sorts of homodimers, or you might say, well, ignore this sequence 5. And so you can basically feed it all these things, and it will tell you what percentage of the peaks are correct or incorrect or unmatched. The other thing that we put in are these new HTML reports, so that for each file, it'll just generate an HTML report. You can quickly open those in a browser or share them with colleagues. And there's buttons to do open all HTML, or you can click a line and open it in UniDec. So this is like a screenshot from yesterday's build. You'll see what the most recent thing is today. But basically, we're trying to set it up actually for a lot of what you might do, of like, if I run like a 96-well tray of results, could I just feed in a spreadsheet and have it analyze all the data for me, and then allow me to like kind of scroll through the results and look at everything? So yeah, and this is kind of what the new report format looks like. We're just dumping like tons of different things, and we can look at more what that looks like. So anyway, it's currently available. It's on PyPy, it's on Docker, it's on GitHub. The UniDec 6, I haven't put up the compiled version yet, in part because I'm still working on the adding new features to the UPP. But if you want a version, definitely let me know. But you can pull it off the V3 branch, the testing branch that I use. So basically, that's the nightly build. Feel free to just grab it. We're working, there's some other cool new features in version 6. So I've added new site matching. So like if you want to basically say, we've always had tools to match, to say like the protein could be bound to this number of things, and you can mix and match different things. But sometimes you may want to say, particular for PTMs, like this site can have these potential PTMs, and this site can have these potential PTMs, and it will generate now the mix of all those possible combinations. So cool plotting options, some new speed improvements. So we're working on basically finishing up the up and seeing where it goes. So with that, I'll thank my wonderful group and all of our funding and collaborators. Definitely the place to go is take a look at the GitHub site. We have a pretty extensive UniDec wiki page that's always expanding. And so like I have all of the hidden features in there. There's lots of little tricks that you can play. I'm always happy to get emails and hopefully answer pretty quickly. So that's the scheduled portion of the talk, and the rest, basically, we can do whatever you want. So what would be useful to see?
Well, I think some of us already know it, but I guess maybe it would be awesome if you could walk us through like how you just deconvolute like a simple native MS spectra. And then I did not know that you can overlay this stuff. So that would be very interesting for me to see how you do that with like the double deck.
Oh yeah. All right, let me get out of this. Everybody can still see my screen, right? So let me start off with Unichrome, actually. This is our chromatography viewer. Just launching this now. For any who are curious, I'm a huge fan of PyCharm, so I just launch it all out of PyCharm. Okay, so this is Unichrome. It's not beautiful. If you want to clean it up, you can. I definitely it's mostly me that develops this, but I am always happy to have new developers and other people that want to put stuff in. And I get a lot of suggestions from people, and that's why it's been useful. So, all right, so here's the data that Daniel sent. Maybe I'll just drag this raw file in, and you can see what it looks like here. This is now the tick, and so we could go through and just select a region here, right? And then if we open selection in the UniDec GUI, it'll just pop open UniDec and we can play with it here. So this is the data. We can just click run and roll the dice and see what happens. It doesn't take that long. You can see, let me walk maybe through how I look at the data. So the first thing I look at is down here. And this is the mass over charge versus charge. So you can see we should get peaks where we see peaks in the data. And if it's a regular charge state distribution, we should see like a swoop, right? Because as we go to higher charge, we go to higher or lower mass to charge, right? So we should see it kind of swoop like this. And I definitely see that here. I would say we do see some signal here. I'm not sure whether I believe that. I don't know. Do you think these other things are real proteins in here? Or do you think that's just artifacts from like regularly spaced?
So I would expect some monomers as well.
Cool. So let's go ahead now and pick the peaks. So you can see here, this is the peak detection. We can play around with the parameters for that if we want. You can see down here, it's now showing like all of the different values. And if we want, we could like label the charge states of this so we could see. Okay. There we go. I don't know why it's so slow. Sometimes when I run Zoom at the same time, things get weird. Yeah, so this is a good way to just quickly like see, did I get the right charge states? The other thing you can do is that you can plot like this and it'll separate, or it'll plot the distributions here. Let me go through and normalize the data. There we go. Maybe what I'll do is just go zoom into this region. So if you zoom in, left click and then you right click, it'll automatically set the M over Z range. And you can see this looks beautiful. It's a really nice spectrum. Now you may say, well, what about these other peaks? I want to see what those are. The way that we set the charge states, we just collapse all of those and go to the red panel. We give it a range. Sorry, the way that we detect peaks is that we give it a range. We say it has to be the maximum within a certain range, and it has to be above a certain threshold. So basically right now it's saying only look for things that are the maximum plus or minus 500. So if we lower this to 50, I think it will now get that. Yeah. We're just now looking for like smaller or like peaks that are closer together, I guess. If we want to see some of these smaller ones, we'd probably lower this threshold, maybe put it at 1%. There we go. So now we can see all the different peaks and we can look at all the masses. If we want to look, we can maybe label this and say, what are the differences from this peak, and see kind of like, oh, this 260 may be addition of something. Yeah.
It's great. I'm definitely learning some new tricks here. How did you do the labeling again?
Just right click on any peak and then say display differences from. There's a new one also, display consecutive differences, which shows like how far apart each peak is from the next peak. But yeah, if you just pick one and say differences from, it'll pop that up and show. I don't know why the GUI is updating so slowly. I think it's a Zoom thing. Normally they just pop in, but it's doing them one at a time. I don't get it. Yeah, and then there's also things like you could label the masses, or you could, we'll show you how to label the names, but you can put in the names later. What else? You could also get the centroid or the uncertainties, like the full width half max, or the error between the different charge states. So all of that's in here. You can also color the peaks like this if you wanted. So there's a lot of different ways that you can like visualize, and it's really easy to add stuff in if you want me to add in like, hey, like someone was like, hey, I really want to be able to label the intensity on the peak. And I'm like, okay, we can do that. That's not hard. So all of that's there. The other thing you could do is like, if you want to copy some of this information out, you could open up, I'll just open up Notepad because it's faster. You just right click, copy all, and then you can see it'll paste that data in. And if you paste it in Excel, this will look like a normally formatted table. But the other thing to know is that in the background, if you go to advanced and go to save file directory, it's actually producing lots of different text files that you can import and do whatever you want with. So like you can see we have peaks, which is just going to be like all of the masses and intensities. We have things like the mass, which is just, this is our deconvolved mass spectrum. So basically there's lots of different stuff in the background that you can dig up and plot in different ways if you want to make some custom plots. And if you're curious what all of those are, I've to some degree explained them. I'll just briefly open up. If we look at the UniDec wiki page, there's a lot of different things on the wiki page, and like a lot of the things that we're talking about on the peak panel are kind of explained in here about how to do things like this. Yeah, this is an example of what it looks out when you nicely copy paste it into Excel. But one of the things in here is what are all those file outputs and how do I export my data? So this is a lot of explanation of like what each of those files do and what they mean. And so if you want to look it up, it's all there. Cool.
Can I ask another question before you move on? So it's very at the beginning, very fundamental. So you dragged and dropped in the raw data, and I think I shared like the profile mode data with you. Would it make a difference to like use Centroid instead, and especially with regards to efficiency?
I think Centroid data would probably be fine. I think it's going to plot it as profile data though, so there could be issues with that. I don't know, actually. At one point I tried to make it much more tolerant of sparse data, and so I think it should work, but I will say I haven't extensively tested it. So
especially if we would want to like deconvolute like full like LC runs, right? I guess getting like the less dense data might speed up things.
Yeah, I think it would work. Yeah, it'd be fun to try. Yeah. Cool. So maybe let's go in. Let me show off some of the peak matching tools because I think that's really useful. So under tools, you can go to oligomer and mass tools. And what this is now is there's a couple aspects of this window. The main one I want to focus on is the oligomer builder. So you said this is a hexamer, and I'm guessing like we could probably calculate the monomer mass. We'll just take a guess.
So heptamer.
Heptamer, sorry. So then 157, 390 divided by 7. So let's say, yeah, actually, hold on. If we zoom back out, let's cancel that. Let's go back to the full spectrum because I think I saw the monomer in there. Now we need to maybe increase our threshold a little bit. We're getting some noise. Okay, so let's assume 22,550 is the monomer, right? So what we can then do is go to tools, oligomer mass tools, and let's put in the monomer mass as 22,550, and then say we can have up to 7 monomers, and that was what, clip P, right? So now if we maybe limit let's just go ahead and click match to mixed. So you can see now what we're getting. It says this is the peak, this is our match, this is our error. And you can see this is 1, 2, 3, and then 7. Now, some of these are matching well, and some of these are not really. So maybe let's limit it to like within, let's say 50 Daltons. So now you can see these don't match, right? But what we can then do is start to look at the errors and start to say, well, what can we find? Maybe we could sort this table of common masses. We don't really, all right, so we could say maybe there's some methionine loss. And so if we right click here, we can add it to the oligomer builder. And now it's going to add in methionine loss as a variable modification. Maybe let's say, like maybe that's a bound colate. I don't know. If we match now, you can see this is matching to a bound colate with the protein, right? This one's still not matching. Maybe we widen our tolerance to 100 just to get something. And now if you hit OK, you can see it's going to update the names here. So you can now then take these and say label all names, and it'll show you on the plot what the different things are. And so you can load your own custom mass lists. You can load other things. You can put in different, like if you wanted to load in a table of small molecules that you're looking for, you could put those in. So any questions on that? Basically, it's just going to mix and match like each of these lines. So you say it has to have 0 to 7 of this. it has to have 0 to 1 of this, 0 to 1 of this, and it's going to create all possible combinations of those things, calculate the masses, and see what you get. Cool. So far, so good. Awesome. There is also, I'll show, this is a relatively new feature. You can check for alternates, and if you get a green, that means there's no alternates. Maybe let's add in a few different things. I don't know, we could put in sodium and I don't know, say like up to 10 sodium. Actually, that's a double sodium. We match to mix, check for alternates. Now you can see a lot of these have potential alternates. And so you can view the alternates and see what might make the most sense. Yeah, so that's another. And if you wanted, you could select this and that would be your new selection. So there are ways that you can look and see, are there other things that would match this? But yeah, that I think looks better. Cool. What's most useful to see next? Maybe the chromatography features. Sweet. So I showed that we pulled this data in as a tick and then like selected a specific region here and then opened it in UniDec manually. We could also run it in this window, right? And that would just show the results here, and you can do limited sort of plots here. It doesn't give you all the full scope of things, but you can basically do very limited sort of analyses, like drag it, deconvolve it, see what you get. We could drag specific windows and then add them over here, or we could parse it in different ways. So maybe let's do parse it in steps of half minutes. And so we would get like this. So you can see it's chopped up all of the different scans into half-minute chunks. Let's delete this last one because my guess is it's not going to look very good. There we go. And then we can basically take this and do our deconvolution. It'll take a little bit longer because it's doing more scans. This is now our stack data. We can then extract our peaks, and you can see this is the extracted data. There's a lot of different ways that you can extract. So maybe you want to look at peak heights, maybe you want to look at peak areas, maybe you want to look at local maxima. So if you do this, you're going to see this is actually, let's turn off the normalization. Now what we're looking at is like, this is an intensity, it's actually the local max. Although it's weird, I think I need to put in a window here. Yeah. I don't totally know why. So what?
From your experience, what's like the maximum, like in chromatography lengths or number of scans that you would recommend to use here? So can you use like a 2-hour sec run or something and deconvolute it all?
So you definitely could. The question is going to be not so much the number of scans in the data as it is going to be the number of, because each of these is averaged over the course of a certain time point, right? So if you did a 2-hour run, but averaged it over every minute, totally fine. Like 120 scans would be no problem. If you did a 2-hour run and you had like 1 second scans, it may struggle. But honestly, we've done over 1,000 scans before. And it will work. It's just going to take, time for it to deconvolve like 1,000 scans. You can optimize that in different ways. So like you can play with a lot of these parameters to like reduce you can do data reduction or intensity thresholding to try and get rid of some extra data points that you don't need. And that'll really speed it up. Actually, I didn't mention this. I should have put this in the presentation. We're working on developing an imaging workflow. It's based on the same thing. And for the imaging runs, we have like, I think it's about 1,200 to 2,000 scans. And they're basically, we just use some clever thresholding and things like that to just like cut out some low abundance stuff. And it works pretty quickly there. Let's try it. So I have some.
The imaging is from DESI data or?
Yeah. And basically we just read it in and it's in meta UniDec. So it just pretends it's the same sort of thing. We just have an X position, a Y position, a series of scans. We just deconvolve it and then reassemble it based on the position data. This is your data here. This is interesting because you can see this is the one with the drug bound. So it actually does look different here versus here. You definitely get more monomer. I'm not sure what happened in the run. So this is a case where you may want to like select this we could open this in the GUI, run it, pick some different peaks. And then this is where potentially we could go in and put in our monomer. And you said the drug was 719, right?
Yep.
And I forget what the name was.
ADEP.
ADEP. Say we have up to 7 bound of each, and then we could lower this threshold just a little bit. Yeah, so you can definitely see there's, that's your protein. This is your protein with 1, 2, 3. This other peak is just noise. This would be a good example because your APO spectrum had like a series of peaks with different PTMs. It'd be a great opportunity to use double deck. Do you want to see how to do it? Cool. All right. Sorry, I'm kind of nerding out here. So I'll just open up this. The way we would do it, really simple. I just want to get a super clean template spectrum. And that's what I'm going to do here. Let's go ahead, normalize the data. I don't know if it's going to make a difference. It does make a difference. That's interesting. I don't totally understand that. I'm going to zoom in and really narrow the mass range. So we'll do 156 to, let's say, 159, just so we get a really nice, tight, clean spectrum. You can see there's a little bit of baseline there, but that's okay. Okay, so we're done with that. Call that our template. Let's go back here. Let's get this. Open it back up in the GUI. Now, let's just run a deconvolution to see how it looks. Looks pretty good. I might go ahead and limit some of these higher charge states just a little bit. So cut it off at 160,000 again, just to clean it up a bit. Actually, I think we can, sorry, let's say 162, 165. Let's be, yeah. And then here, let's set this at maybe 150. Cool. I think that looks good, right? Now we go under here in the advanced deconvolution parameters. We just need to select our kernel file. And so to do that, we're going to go to the data, open up your example, and then we'll select this one, which was our file that we opened before. This is just the text file that it produces. So when it converts from raw file into UniDec, it just writes out the text file that's averaged. We can also probably select the raw file, and I think it will work, but I'm just going to select this for comfort. And then I turn on double deck here and then run it. Please work. It looks beautiful, right? It's always nerve-wracking doing live demos because you never know when you're going to have a Microsoft moment and it's just going to fail. So I think I'm happy with that. So now if we do Control M, we can match against the things that we have. I think, sorry, let's make sure that, oh, sorry, we lost our things. 22550. This was our P, and we had the other one, which was 719 up to 7. Match to our mixed, lower this down to maybe 100. And you can see we have like the same consistent error on everything. So maybe that's like a, I don't know, some like salt binding or something like that. Or maybe it's just that this was rounded slightly off. UniDec will round to the nearest, let me just go ahead and click OK here. Mass sampling. So we've sampled the mass every 10 Daltons. So it is rounded the nearest 10 Daltons. So it could be off by up to like 4 or 5. And when you multiply that by 7, it's about right. So anyway, now you can label those names and you can see all of your different things. And there's even the fourth bound here, which would maybe struggle to see confidently if you didn't have the double deck in here. And we get that if we now lowered our threshold to like 0.01. I think we would get it. Raise it. See if I can clip that. There we go. Control M will now match them. And now we can label them and see all the things. So.
So a question for the visualization here. So what I see is mainly like the bound versions now, and then yeah, there's the APO too. Can you like highlight which comes from which file? So especially when you have like this control treatment.
Oh, sure.
Situation.
Yeah. So that's where. So if you wanted to stack these, there's a couple ways you could do it. So one is you could load it in meta UniDec, right? And you could create a meta UniDec file with each of those as different scans. But honestly, the faster way to do it, if you run the UniDec launcher, you go ahead and get the launcher up here. There's a tool called the data collector. And if you open up the data collector, you can set a directory, which we'd set probably like this. We could add files. So we can add this one and we can add this one. Actually, there's a bit of an issue with the fact that we opened this directly out of meta UniDec, or sorry, out of Unichrome. So let me go back just real quick. Actually, sorry, I think it's fine. As long as I do this and then add the other file, it's actually hidden in here. This one. Yeah. So we can extract the zero charge mass spectrum like this, and it will overlay the data or show like a 2D grid. Sorry, I can't see you all there, but anyway, this is now showing basically these two files. And if we wanted, we could like play with them in different ways and then click re-extract and update it. You can also do KD fitting. So if you're doing like a series of increasing concentrations, you could get KD values out of this. Does that answer your question?
Yeah. There's of course another way that we would do it. That's like export the spectra as SVG and throw it into Illustrator. So is there a way to like get a SVG out of UniDec?
So in general, most windows will have like a file save figures as PDF or save figures as PNG. So if you look at the main window, you know, like if we have these tools to generate different sorts of figures. The other thing though, and this is a hidden feature that everyone should know about, if you hold, actually, sorry, you just middle click on any plot, it'll open a save figure dialog. So middle click, and then you could say like figure.svg. I think it will save that. Let me go back here. Sorry, I think it's under here. Nope, sorry, hold on. SVG may not be supported. Let me see if I'm getting an error. Oh no, I just am looking in the wrong place. When I looked at the background, I figured out where it was. It's in here. Yeah. So then you could open this and it's going to look like, you know, like you'd expect.
Nice.
So yeah,
it's all vector, right?
So what, sorry?
It's all vector, so it's not pixeled, the trace.
Okay.
Yeah. Perfect. And it was just also like in a main UniDec window, you just middle click and then save as.
Yeah.
Okay, that's awesome. I was like looking for that feature for ages.
Yeah. You can also save them all under the file menu. So you do file, save all as EPS, right? It'll generate that. There is a save figure dialog where you can tell it the size and the transparency or whatever. But yeah, middle click is the easiest. The other thing, honestly, like for people that really know Python, even at an intermediate level, like it's really easy to just go through, grab this mass.txt file in like 3 lines of code. You could plot that in R, in Python, in whatever, right? In Excel, you can plot it. And so you could just get your 2 mass files and overlay them, and then you could put whatever sort of things you want, you know. Yeah. It's kind of the whole ethos of UniDec is trying to put it all, you know, like we're not hiding any of your data, right? It's all there. And if you can't get to it, like it's easy to just put in a print or a dump or something like that. It should all be there.
Yeah, that's super cool.
Yeah. How about checking out some of the chromatography stuff while we're at it? Let's go back, reopen UniDec CD. I'll show some examples. This is some stuff I got. Oops, sorry, that's the CDMS data. This is Unichrome. If we go file, we can load some example data. So here's some, it's native sec with Herceptin. And you can see what we've done. This is our tick here. We've just said start from 4 minutes, go to 6 minutes, and then partition it every 0.1 minutes. And that gives us about 20 spectra. And so, I mean, we can show you how fast it runs. Takes about a second to run all of those. We can probably speed it up if we add in a little bit of intensity thresholding, or maybe even put in some data reduction. Yeah, so you can see when we start doing data reduction, it'll start to clip off some of these plots. And now it should run a little bit quicker. Yeah. You'll notice like the purple one, it mostly eliminated, right? Because it just didn't find anything that was above the threshold there. Yeah, and then what we can do is, pick those peaks and get out these extracted ion chromatograms for all of this. We can plot these different grids. So this is going to be now mass over charge versus time, and this is mass versus time. The other thing to know about, so, we mentioned middle clicking will get you the figure. I believe it's Control U. Yes. Okay. If you do Control U, you could say data.text, and it will save the data from any one of those figures into a text file. So this is just XYZ of that plot that you could then extract and do feature finding on if you wanted. So yeah, there's a lot of little tricks like that, and they're all in the wiki. That's honestly, I forget them a lot of the time, and then I have to go to the wiki page to remember what the shortcut was. So
that is super cool. Can I ask you something about the sec? As this is something I'm also getting more and more interested in. So how does it like perform with mixtures? So if you would have like a bunch of proteins, did you use that before? How's your experience there?
I'm trying to think. I definitely have looked at some data sets like that. It does work, and it should work well. It depends, of course, on how crazy your data gets. But we don't do a lot of SecMS. So like honestly, a lot of what I get are other people that like send me data, and like it kind of gets built around that. So try it out and see. It's all there. So it's just going to go through and parse it, slice it out, and deconvolve it. One of the things to be aware of, there is an experiment. So what this does basically is it sums up the entire trace, and it picks peaks based on that, right? And then you can extract all the different things from it. There is a way that you can do per scan peak picking, get scan peaks. That may be more useful in certain circumstances, but it's kind of up to you to see. The other cool thing, while we're in the experimental menu, is that you can do, if you're acquiring data, you can have it automatically refresh, or you can refresh at different times. So if you're like putting in a file that's under acquisition, you can have it like automatically refresh every 5 seconds, and it will just like keep updating. It's a tool that is pretty crude, and if you find yourself using it a lot, let me know if there are tweaks, because we could build a little window to time it in specific ways. But it's kind of useful for when we're like on the instrument and trying to tune things. It's useful just like click refresh, and it pops up, and then you can like adjust it. So
do you think it's like that's an avenue that more and more people are using, like sec for native MS?
Yeah, definitely. I wish we had it set up more reliably in our lab. We're working on it, but it's the problem is for our nanodiscs, it's like kind of hard to ionize with the normal Hezi source. And so we've been trying to figure out other ways to do like really reliable ionization, because with antibodies, they're really robust and anything will work. But it's a little bit harder with nanodiscs. So
there you normally use nano ESI.
Yeah, just pulled needles.
And then all the sec is done with like normal flow, I assume, or microflow.
Yeah.
Do you have any recommendation on columns, by the way?
No. Yeah, I'm not the right
people use.
Yeah. I ask around. Yeah, I think there are a handful of, I think they all, any biosec column should work pretty well, I think. Yeah. Cool. What else would people like to see?
Can also be very basic.
Yeah.
Kind of like nerded out a little bit as well. So if you have
I guess
very basic questions, feel free.
If there's anything interesting you want to like, there is this tool here you can do. If you want to look at the grids in here, you can animate them. So like if you want to look at the M over Z versus charge grid, you can like click through here and you can click through the different time points and it will show you the grid. Sorry, I need to zoom out. There we go. It's not super exciting. You can see there's not much changes. But that is possible if you're interested in that. You can even make it play a movie and put on a certain frame rate. No promises it's going to work when I'm on Zoom. I guess maybe I'll lower the frame rate. Yeah, so you can kind of like skip through your data that way. You can also do the same with like spectra. So if you want to just look at the like zero charge spectra, click play, auto scale it. Yeah, it'll show you all the different spectra. What else is cool that nobody knows about? You can batch run with this. So if you set up like a series of things, you can batch run it. Manual assignments are really useful. That's something not a lot of people use, but if you have a really complex spectrum, let me go back to just regular UniDec because that's easier to see. If we just pull in some of your data here, let's pull in this file. We can go ahead and like deconvolve it, right? Let's say that we now know that there's some potential overlapping charge states like here, right? One way we could do it is we could go through, pick the peaks, label the charge states so that we can see what they are. And if we wanted this to not have any dimer in it, we could say, select that little, oops. I don't know why it's zoomed out in a crazy way. Oh, it's because it's got this. We could go to manual assignments, zoom into this peak here, right? And then we could add it from the zoom range and tell it that's a +8, right? And then hit OK. And then turn on under here the manual mode and run it again. And you can see this little blip should disappear. We've killed a lot of the dimer signal because we forced that to be like a +8 charge state. So that's useful if you have like really complicated spectra and like you need to go through and manually assign some of the peaks, and like UniDec is just struggling with it because there's too many possibilities. You can actually go through and kind of force different things onto it. You can also force it to only go in like a window around allowed masses. So if you click this, you can put in either like a list of allowed masses. So let's say we did like, we have our 22,550 up to 7. We could say these are all the masses that I will allow, and then hit OK and say only allow peaks that are within, say, 5,000 Daltons of each of those allowed masses. And you should see, yeah, it's going to clean things up. You're going to get some artifacts that get squished into here. So I don't know if I believe all of this. Oh, I should probably turn off manual mode. Maybe that will help. Yeah. Anyway, so like it is starting to fit some noise into each of those ranges. So I don't know if I would trust it, but it is a way to kind of limit what mass species are available. Yeah, there's lots of crazy stuff you can do. I don't know. Yeah.
That's all super cool. Yeah. Can you write MZML files after deconvolution?
No, but it wouldn't be that hard to do. Like I think that would be a few lines of code to add in. So if that's something that you really want, we could talk about it. We use mostly PyMZML as the library, and they don't have a writer built in. But I know that OpenMS does. And so we have used PyOpenMS for scripts and things. So the only downside would just be that would require PyOpenMS now as a dependency, but we can make that optional.
So one thing that I could foresee for us is that we make use of other like feature finding tools downstream. So especially working with like the guys from MZMine, I would love to deconvolute and then pull it into to MZMine also.
Yeah, I think, and you know, we could also write a script. So I guess I prematurely ended the screen sharing. We have, I think I mentioned, we have all of the data in those HDF5 files. Like here, I'll share here. This is what the HDF5 file looks like. So we have the config object, which has just a bunch of flags as the metadata. But then each of the scans basically just gets its own folder. And within each of theirs, there's the mass data object. And that's just your mass spectrum. But then also each of those is sampled slightly differently. So then there's the mass grid and the mass axis. So this is the X values and then the grid of Y values for basically like time versus mass. So we could write a script to pretty easily convert this HDF5 file into an MZML. Yeah, you could then basically, it would be like a deconvolved, like zero charge mass versus time. That's a cool idea.
Yeah. So you used to do that with extract and Excalibur.
Yeah.
But they somehow discontinued that. And then in the new, what is it, like freestyle, extract does not support that anymore. So I think that's a real niche. That would be awesome if you could fill that.
Yeah. I think we could do it. I'm going to put it on my to-do list. Yeah. Cool. Yeah, and I mean if you just wanted to write like a single spectrum, that would be like a few lines of code, right? Just read in the mass.txt, you know, parse the array, stick it into the MZML file and write it. So
yeah, I think most interesting it would be for again like LCMS data.
Exactly. Yeah. Yeah. Cool. I like it.
Awesome. Well, thank you so much for all this tips and tricks and awesome intro to UniDec. I don't know, are there any other questions?
Right. My email's online, so I really do like getting emails, and I would say I get probably 3 or 4 per week from people asking about how do I do this, or could you add in this, and like a lot of these features got added because somebody needed it and just asked. And I'm not a computer scientist, but I am learning to program in much more flexible ways. And as the code has grown, it's gotten easier to maintain and easier to add new things and much more modular. So like, it's pretty easy to add like simple new things, like this idea of like, could we write it to MZML? It's like, yeah, I think that would be probably 10 lines of code to figure out. So it's really like, it's not too hard to do a lot with this. And I'm always happy to get suggestions.
Awesome. All right, then, yeah, let's thank Michael again for this awesome talk. And yeah, thanks all of you for stopping by. And I hope to see you next time in 2 weeks.